Thanks everybody for attending today's session.
Thank you for the introduction. Hello, everybody. Thank you for joining us for this discussion. This presentation will focus on the methods employed to minimize the number of animals used in toxicology studies while still ensuring robust and reproducible findings. Drawing from Altasciences' extensive experience spanning decades, we will analyze the advantages and limitations for each method. As we all know, it is essential that the scientific community continually reevaluate study designs and technology that will enable a reduction in the number of subjects used for toxicology studies, an approach that aligns very well with the 3 Rs of experimental animal welfare: replace, reduction, and refinement, but also keeps the overall study objectives in mind. On the agenda for this meeting, we will cover the following topics: the relevant testing models, what species are required by various drug modalities, microsampling. For several studies, Altasciences has adopted blood microsampling as a refinement to the more common needle and syringe technique by leveraging the Mitra volumetric absorptive microsampling device for accurate and precise collection of low sample volume. We will cover examples of some study design adjustments which allow us to retain scientific integrity but reduce the number of animals used. As always, with each choice, we have the positive and negative sides. Therefore, we will briefly touch upon the pros and the cons of each proposed method. And we're going to wrap up the webinar with a summary slide to quickly talk about all of the discussion points mentioned above. So let's get right into this. When are the non-human primates the only model relevant to your research? Well, when you're working with antisense oligos, gene and cell therapy drugs, monoclonal antibodies, and even some select small molecules. Given the wide variety of drugs that would require the use of NHPs, we're faced with the current challenges of limited animal supply. Therefore, we as an industry need to challenge the status quo and come up with innovative approaches to maximize animal use. Later in the presentation, I will briefly speak regarding the use of juvenile animals of varying ages. So let's get right into this. The first approach is aimed at an animal reduction in small animal studies with the use of microsampling. Microsampling strategies allow us to consolidate satellite toxicokinetic and main study groups in an effort to reduce the overall number of animals used on the study. Additionally, this method allows for toxicological effects to be correlated directly with an exposure profile in the same individual animal. Capillary microsampling techniques also circumvent the hematocrit effect often reported for dried blood spot analysis. However, the processing is tedious, and drugs exhibiting nonspecific binding or requiring matrix stabilization are problematic. A recent alternative is volumetric absorptive microsampling, wherein an accurate blood volume is absorbed onto a hydrophilic polymeric tip, simplifying sample collection. The use of this technique allows for a dramatic decrease in the number of animals used in a tox study. A pharmacokinetic rat study can successfully be completed with 3 animals per dose level via serial collection, and a PK mouse study can be completed with 8 animals per dose level, split into the 2 subsets using the sparse method. This represents a reduction in the population by 50 to 60% respectively, compared to the study designs that use a more standard sample volume of 0.5 mils per sample. What you see on this slide is the Mitra microsampling device or kit, which includes the following. It has 4 sampling tips, which allow for collection of 10, 20, or 50 microliter samples, the clamshell holding case for the tips for storage and shipment, which allows for very fast and convenient packaging of the samples, and virtually no drying time is required when storing them properly in the provided bag with a desiccant. The shipping bag with a desiccant is also included as part of the kit available for use. So briefly about the sample collection and handling. So As you see on the slide, the collection area is shaved and it's wiped with an alcohol wipe. A small hole is made with a needle. The first drop of blood is typically wiped off, and then the second drop is allowed to be absorbed by the tip of the device until the tip is fully red. Pressure is then applied to the collection site to allow for hemostasis, and the sample collection process is complete. The benefits of this method are multifaceted. Not only are we able to cut down on the number of animals used, which I will go into a little bit later in the next slide, but this method also does not require extensive technical training. The procedure is very fast, so the animals are not restrained for a prolonged period of time. And there is no requirement for processing, as the tips are shipped in the clamshell holding cases that are provided with the kit at room temperature. So now to go back to the actual point of this webinar, which is how is using this method going to help us to cut down on the number of animals? So let's take a look at the practical benefits of using the Mitra tips and how it relates to the decrease in the number of animals used on the study. Let us use a standard design with 15 time points for complete TK profile assessment. So the pros of using the tips is that in the routine study with terminal blood collections, now we're talking about the CD1 mice, we would need to use a total of 122 animals for a sparse collection with 3 animals per sex per time point. With using the tips, the number of animals could be reduced to a total of 56 animals. We'll still be using the SPART collection method, but now we'll be using 4 animals per sex per time point, but we're cutting down the number of animals by 50%. The cons of this method is that a new bioanalytical method may need to be developed and validated in order to utilize the dry blood collection technique. Now, looking at a typical tox study with a Sprague Dawley rat, again, we're going to take a look at a potential of 15 time points that are needed for a complete TK profile assessment. So in a typical study, you would need a total of 90 rats, 3 animals per sex per time point. However, with using the tips, we are able to completely eliminate a TK subset because the blood volume is so small, and we can collect those blood samples for TK analysis from the terminal tox animals using a much less invasive method, thus eliminating any potential complications associated with jugular blood collections, which is the routine method for TK sampling. The con of this method remains the same, that a new bioanalytical method may need to be developed and validated for the use of the dry blood collection technique. Now, switching gears a little bit and taking a look at the modified large animal study designs. So let's take a look at a cynomolgus monkey study. So a typical study design for a routine tox study would require 4 dose levels. Typically, if you have a recovery period, you would have a terminal subset with 3 animals per sex per group, and then a recovery subset with 2 animals per sex per group. In the particular study that I'm using as an example, the control group animals were administered saline. Because saline is so well defined and characterized, there is no need to continue evaluating the animals throughout the recovery period because there's nothing that they would be recovering from. In addition, in this particular study and in this particular case, the test article was very well characterized, and we knew that at the lower dose level, we did not anticipate any test article related findings based on all of the previous studies that we have done. Therefore, again, there was no need to have a recovery subset. So with using the previous information that is available to us, we were able to eliminate 4 animals per sex per 2 groups from the study. So a total of 8 animals. So obviously that is a huge plus because these are monkeys. Everybody knows the monkeys are quite pricey. So eliminating 8 animals from your study design while still retaining your scientific integrity is great. The cons here are that if for whatever reason in the new study that you're conducting with maybe a more extensive dosing period, there's a newly developed test article related change that is identified in a low dose group, you would not be able to track it through the recovery period. Also, in this case, the control article was saline, so it is very well characterized, and it's not always the case for the tox studies. So that is a big drawback as well. And to counteract that drawback, a robust historical data set for clinical and anatomic pathology would be invaluable to have an age-matched animals for your data evaluation. One other design modification that I wanted to discuss does not necessarily address the reducing number of animals in a particular tox study, but it does address the issue of repurposing their use. So in this case, we had a tox study with a typical study design, again, with 4 dose groups, control, low, mid, and high, and the animals were administered the test article via an IV infusion twice, I believe. This was a gene therapy drug, so the sponsor was interested in figuring out PK and tissue distribution within the eye. So what we have incorporated into our tox study design was administration of the test article via an ocular route about 24 hours prior to terminal necropsy. And then the eyes were then collected at necropsy using specialized media and shipped to the client for analysis. The advantage of this approach is that we're using and repurposing existing animals and we're eliminating the need for a standalone PK or biodistribution study, thus reducing obviously the number of animals used and also the overall timeline for drug development, as well as cost, considering again the price of the non-human primates. An additional benefit is that in this case, we were then able to compare the ocular and peripheral PK profiles within the same animal. And more recently, in the more refined version of this design, we have administered the test article again via the ocular route and are monitoring the animals for a period of 1 week prior to necropsy. So now let's take a look at the juvenile NHP studies. The need to use the younger animals, typically less than 12 months of age, presents several challenges. One big one is the transport of animals between the supplier and the testing facility. If the animals are too young, they may need to be either transported with their mother if they have not yet been weaned, or they may not be able to be transported at all, and the breeding will need to take place at the testing facility. As I'm sure everybody's aware, the breeding process is very complicated, very involved, and not every facility is set up to accomplish it successfully. In addition, again, smaller monkeys equal smaller blood volumes available for use. And then smaller size of the animals may also affect the technical aspect, such as dosing, blood collection, IV catheter placement. So Altasciences has conducted a review of the available immunologic development data over a wide range of animals of various age ranges to look for the similarities across various parameters. And the reason we did that was to see if we're able to expand the age range of the animals to be used in the juvenile studies. So we're not stuck at using animals of just 6 months of age or just 9 months of age, but we can expand it and use animals from anywhere from 9 to 12 or maybe 9 to 15 months of age. Again, as close as we can get to larger animals, older animals, while still fulfilling the needs and the objectives of the study, gives us much more flexibility with this collection of blood samples and dosing and a lot of the technical aspects of the study, as well as getting the animals on site for the study. By expanding the age range, again, we're able to reduce the number of animals used on the study, reduce the stress on the animals because the younger animals, as I'm sure we're all know, are going to be more stressed when they're handled for procedures. And the additional benefit is also the reduction of the overall study timeline. If you're able to enroll your animals faster, you can start the study faster, you can get your report faster. So let's take a quick look at available immunology data that we have reviewed for this presentation. So to do so, we have looked at the data from animals that were 90 to about days old to about a year old. And specifically, we looked at flow cytometry and T cell dependent antibody responses. As you can see on the slide, the CD3 T cell population in animals of varying age groups from 90 days to about 360 days is very consistent. And that's across both males and females. When looking at the B cell population, we're seeing the same picture. The data is maybe a little bit more variable, but overall it is still comparable. And again, there's not a lot of variations that we're seeing between the males and females of animals in varying age groups. Taking a quick look at the TDAR responses following a KLH challenge, we can see that both IgM and IgG values are again similar across animals from various age groups. Here we're looking at the IgM responses in animals that are 90 days through 270 days old. And we see that in all of the subsets and subpopulations, there is a rapid elevation following the primary and booster doses, and then a sharp drop on subsequent days. When looking at the IgG levels, we see a gradual elevation after the primary dose and then a more rapid increase after the booster dose. And again, the point here is that the immunological responses are similar and consistent across age group. So we're well set in expanding the age range of animals to be used in the study and enrolling slightly older animals on the tox study. And to briefly touch on the study design modifications in dogs, it's a little bit similar to the options that we would have in the routine tox study in NHPs. In that, again, we have to leverage the information from previous studies in order to see if we can reduce the number of animals in the recovery subsets. So in the example I'm using here, we're eliminating the recovery subset from the low dose group because again, we've set the levels, dose levels in such a way that we know that there are no changes expected at that dose level. So there's no parameters that we would need to track through the recovery period. And again, having the robust historical data set is critical when employing this approach. So to conclude our talk, a couple of points that I wanted to bring back to your attention. The use of Mitra tips significantly reduces the number of animals required in the routine rodent toxicology study. Microsampling, which is much less invasive than a traditional approach like a jugular bleed, has the additional benefit of eliminating potential complications such as death or histological changes. Collection of TK samples from the main tox animals allows for the correlation between the possible test article-related effects and the exposure levels. With careful consideration, large animal studies can also be designed in such a way to provide robust and statistically significant data sets while reducing the overall number of animals used. Accurate PK studies with no imaging can be eliminated and the assessments can be included in the toxicology study. For NHP juvenile studies in animals between 12 and 15 months of age, the immunological data suggests that there are no major differences in the responses. Therefore, you can expand your enrollment age of the animals and so that you can cover a wider range of corresponding human age ranges. We at Altasciences are actively working on creating a robust background data sets, which may be included in the future as virtual controls. As I noted in the beginning of the presentation, it is absolutely essential that the scientific community continually reevaluate study designs and technologies that will enable a reduction in the number of subjects used in tox study. All these approaches align with the 3 R's of experimental animal welfare: replacement, reduction, and refinement. Thank you so much for your time.
Thank you, Noreen. Very nice presentation. So at this point, we will entertain any questions. Some questions have come in and we'll just go through them. First question, are you concerned about the statistical power in non-rodent studies?
So I think that we can all agree that robust statistical power on a large animal study always presents an issue. In most of the studies, like let's take the most routine tox study we've discussed before, we would have 3 terminal animals per sex per group and 2 recovery animals per sex per group. So already we're in a position where we have just enough animals for statistical calculations in a terminal subset, and then a little bit less than that for the recovery subset. However, in the cases of the studies that we evaluated at Alta based on the updated study designs, we didn't find that there was an impact on the assessment of the study data. It was less than ideal in terms of, again, the overall statistical power, but that did not impact the interpretation of the study.
Thank you, Noreen. Another question. How widespread is the use of microsampling in rodent studies?
Unfortunately, it is not as widespread as we would like to see. One of the limitations that I have brought up during the presentation was the need to develop a bioanalytical method when a traditional method might already be available. So that is certainly a challenge because that does require additional resources and time on behalf of the sponsor. However, our colleagues in Laval are ready and able to work with our clients in developing these unique methods to allow for a much improved study design.
Thank you, Noreen. I see another question. Does microsampling also transition to the clinic from preclinical studies?
Good question. I believe so, because it can certainly provide patients with a benefit because the amount of blood that is needed is so small. It would be akin to folks that have to check their blood sugar on a daily basis, for example. It's a tiny, tiny prick with a little needle with just a drop or two of blood that you would need versus having to collect a mil or 2 mils of blood using the traditional method. So I think the transition to the clinic of this method is a pretty good idea.
Another question, what percentage of studies were GLP and intended for IND submissions?
So I'm assuming the question is in regard to the updated study designs that were described in the webinar. And all of the studies that for which the data was reviewed were GLP studies. Most of them were IND enabling studies, but all of them were certainly used in the package for the IND submission. So in some of the cases, there was actually a pre-IND meeting that was held by the sponsor with the FDA, who have looked at the proposed study design and then approved it prior to going into the preclinical study. And to date, we have not heard back from any of our clients regarding rejection of any study data or any concerns from the regulatory agencies in regard to data evaluation based on the updated designs.
Thank you, Noreen. Another question that has come up is that you mentioned difficulties with obtaining young juvenile animals to fill a study. What could some of these challenges be?
Thank you for the question. The challenges with obtaining younger animals, which for the sake of this discussion are those that are at or slightly above 6 months of age, are really multifaceted. First, one has to consider how to get these animals to the testing facility. In order for the animals to be enrolled at a study at 6 months of age, they would need to arrive at the testing facility at least 1 month prior to the start of the study. And that's to allow them to undergo domestic isolation and then an acclimation period prior to dosing. However, the typical age of weaning the animals from their moms is about 6 months old. So that means in order to fulfill all of these requirements, pregnant dams would have to be shipped to the testing facility and be allowed to deliver there. And then the infants will need to age up basically to the appropriate age of 6 months old, because obviously we don't want to ship the dams with their newborns, because that's a lot of stress on the animals. But even shipping the pregnant animals also adds a complication, because then you would have to select or pre-select more animals that you need for the study, just to account for the potential loss of pregnancy due to the stress during transport. So to avoid these issues, a facility may choose to breed on site. However, successful breeding colonies require a lot of time and a lot of technical expertise. For example, in order to be able to fill studies, you would need to maintain two different colonies of animals. One would be your proven breeder males, and one would be your mensing females. So to select and keep the males, you have to select them obviously based on age, and then you would have to conduct sperm assessments and measure the size of the testes. For the females, daily menses checks are necessary so that you can track their cycles and ensure they have at least one normal cycle prior to allowing them to breed, so that you can find the perfect time, the most optimal time for breeding. So again, this is a lot of technical input, technical expertise, a lot of time and money for the testing facility in order to be able to breed on site. So this is why we believe that extending the age of the animals to enroll slightly older juvenile animals allows one to bypass these challenges and fill the study quicker.
Thank you, Noreen. Another question that has come in reads as follows. What are the technical challenges with working with the younger animals that could be improved by increasing the age of the animals enrolled on study?
Another really good question. So, as you may know, in the recent couple of years, the age and sizes of the animals enrolled in just routine tox studies has dramatically decreased. Gone are the days where the average age of the toxed animals were 3 to 4 years old, and the average body weights were about 4 to 5 kgs. So the industry had to adjust by being more selective over what samples are collected during the study, and how to refine the methods to run the bioanalyses based on smaller volumes of blood. With animals that are around 6 months of age, the problem becomes exponentially compounded. The animals are on average probably a little bit under a kg, so the allowable blood volumes are severely decreased. In addition, every procedure for these animals becomes even more challenging because of their size. Dosing and blood draws, for example, require more specialized training because their veins are smaller. You may have to rethink the way that you're restraining the animals. We at Alta use routinely the restraint chairs. These animals may be a little bit too small to fit there comfortably and safely. And then we also can't forget the increased distress on these young animals, because obviously we know the stress most often leads to multiple GI disturbances. And because these animals are so young, it is often a little bit harder for them to recover from a bout of 2 to 3 days of diarrhea. For an older animal, for example, it might be a lot quicker. You just put them on pepto-lactofiber, and then they're fine within a couple of days. The situation gets a little bit more dicey when we're talking about smaller, younger animals. And again, this is why we strongly believe that increasing the age range and the size of the animals really helps with addressing these concerns.
Lorraine. Another question. In view of general shortage and pricing of the non-human primates, currently. Are there other species that one may consider in order to overcome this, challenge of the current situation of NHPs?
Yes, so mini pigs are an option. This is more practical now that there is a Sinclair nano pig that has a similar weight to a beagle dog at about 12 months of age. So that means that your test article requirements are manageable. Therefore, we believe that the nanopig can act as a good alternative to use, especially since dogs are companion animals. So there's sometimes some limitations or complications based on that. So I would say that a good alternative for an NHP would be a nanopig.
Thank you, Noreen. I don't see any other questions. However, if you have any additional questions, please feel free to send them to myself, Noreen, or Altasciences. Thank you again for attending this session.
Thank you, everybody, for your time.
Thank you, Noreen and Norbert, for your presentation. On behalf of Altasciences, we'd like to thank all of you for taking the time to join our session. Enjoy the rest of your day.